The Biochemical Society and Portland Press are pleased to welcome you to this webinar, part of the Biochemistry Focus webinar series. Topics in this series include different research areas in the molecular biosciences, as well as practical sessions to support career development. Each webinar will give you the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in our webinar series, so please see the website for more details. My name is Melanie and I'm a research scientist in Australia. You can probably tell from my accent that I'm Australian. I'm located in Melbourne. In Melbourne, I work at the Walter and Eliza Hall Institute of Medical Research, where I run a lab and a division focused on epigenetics in development. Today's webinar is titled Reprogramming the Epigenome Through Embryogenesis. As you know, epigenetic marks are by definition mitotically heritable. However, in contrast to this really strong heritability, epigenetic marks need to be cleared between generations in order to remove the marks acquired during the parent's lifetime. This is a critical process to ensure totipotency and viability of the offspring that has been studied most extensively for DNA methylation, particularly for DNA methylation at CG dinucleotides in mammals. In this case, we know there are two key rounds of reprogramming, one during germ cell development and another shortly after fertilization during pre-implantation development. Interestingly, the embryonic round of reprogramming occurs alongside activation of the zygotic genome, which has been largely silent during the final stages of germ cell development. So this is also a process of key interest. Now, some of these processes, as I said, have been largely studied in mammals, but there are several areas that we're really still very unsure about, and these include the role of methylation at sites that aren't in CG dinucleotides, so non-CG methylation, for example, but also what happens in other species, and we all know that we can learn a lot by comparison between different species. So today we will explore how the epigenome is altered in embryonic development in mammalian and non-mammalian species. From a non-mammalian perspective, we will hear about inheritance and developmental reprogramming of non-CpG methylation in zebrafish, and we'll then hear about early mammalian development from fertilization to gastrulation with a focus on zygotic genome activation and the transfer of the control from the mother, or the oocyte, to the embryo. So this is perhaps, I think, one of the most fascinating and important fields of biology, although of course I am biased, as it really pertains to how much of life is created, and we know that there's still a huge amount that we don't understand based on the complexity of these processes. Our first invited speaker today is Dr. Ozren Bogdanovic, a lab head and senior research fellow at the Garvan Institute of Medical Research and the University of New South Wales in Sydney. He leads the developmental epigenomics lab that investigates the contribution of epigenetic mechanisms to embryogenesis, germline formation, and disease. His lab combines zebrafish approaches, CRISPR-Cas9 genome editing, and genomics to study the developmental dynamics of epigenetic marks such as DNA methylation and histone modifications in vivo. Our second invited speaker today is Dr. Melanie Eckersley-Maslin. She is a BBSRC Discovery Fellow at the Babraham Institute in the UK. She investigates how the epigenome sets up and initiates transcriptional programs in early mammalian development using embryonic stem cells and mouse models. She will soon be moving also to Australia, to the Peter MacCallum Cancer Centre in Melbourne, across the road from where I am, where she's going to start a new group leading, investigating into epigenetic parallels between early development and cancer. So we're going to start with Oz, and then he's going to hand over to Melanie. So welcome, Oz, and we all look forward to your talk.
Thank you. First of all, I'd like to thank the Biochemical Society and Marnie for organizing this fantastic webinar. Today I'm going to be talking about a very peculiar DNA methylation signature that we recently discovered in zebrafish. So as Marnie mentioned, the DNA methylation has been extensively studied during embryonic development. However, this was mostly studied within the context of CpG dinucleotide, which is where most methylation is taking place in vertebrates. However, a number of recent studies have demonstrated that non-CpG methylation, or MCH methylation, so where H is essentially any nucleotide besides the G, can also take place particularly in neurons and in pluripotent stem cells, as well as in oocytes. So while classical CpG methylation is pretty much anti-correlated with transcription, with non-CpG or MCH methylation, this is not so clear. So some fantastic work from Ryan Lister has described a few years ago that DNA methylation is actually anti-correlated with transcription in vertebrate, well, in mammalian brains, and it is positively correlated in pluripotent stem cells. So what happens during mammalian embryogenesis with this non-canonical form of methylation, as I mentioned, it is present in the oocytes, it's not present in sperm, but upon fertilization it gets diluted and it disappears only to re-emerge again during fetal development, as you can see by this purple line. And this happens in both mice and humans in the neural system. And recently, Ryan's lab has shown, and we participated in this study, it's currently under review, that this form of non-CpG methylation in the brain is actually highly conserved among vertebrates, particularly in this CAC context, which is typical of neurons. So knowing that in mammals, both MCG and MCH is globally depleted, and that this does not happen in amniotes, such in fish or frogs, which is something that we've shown before, we wanted to explore whether MCH could be present during zebrafish embryogenesis, and if so, what would be the functional implications of this? So first of all, I'd like to mention that this project was and is being led by Sam Ross, an extremely talented student from my lab, who did most of the work here. So the first thing that we did, we decided to generate genotype-corrected methylome maps, so whole genome bisulfite sequencing maps, for 4 stages of zebrafish embryogenesis, covering blastula, pharyngula, tailbud, and the adult brain. And as you can see on the left side, this is essentially the lambda control, which is what we add to bisulfite sequencing experiments. It is a negative control, so wherever you see anything that's 0.2, 0.4, this is likely just noise in the lambda. So anything within that range should be, taken with a grain of salt. And you can see essentially that there is some enrichment in CH and in particularly in CA dinucleotides that go over this threshold, and particularly in the brain, as shown before. However, when we looked at the top 10,000 most methylated motifs in this non-CpG context, what we discovered was this very peculiar motif, so CAT, GCT, AA motif, which was present in almost identical shape and format at all 4 stages. And when we looked where these sequences were located, where these motifs were located, they were predominantly located in the MOSAT DR repeats, which is a zebrafish or perhaps fish-specific repeat family, mosaic satellite repeats. And you can see an example of such a repeat here with its corresponding DNA methylation pattern, as well as some single nucleotide profiles where black circles denote methylated TGCT sequences and white circles denote unmethylated TGCT sequences. So essentially, embryonic MCH is enriched at MOSAT repeats at this particular sequence. And one other important thing is that we see it almost exclusively on the TGCT strand and not on the opposite AGCA strand, which would be a possibility similarly to CG methylation, which happens on both strands. So next, what we wanted to do was, knowing that bisulfite sequencing is prone to all kinds of sequence biases, we wanted to validate these results by an orthogonal approach, and we applied enzymatic methylation sequencing, which is a technique that was developed by New England Biolabs, and it essentially entails TET oxidation and ApoBac conversion to unravel methylated and hydroxymethylated sites. And what we saw is that the profile was pretty much identical with EM-seq and whole genome bisulfite sequencing. So the MOSAT sites with this particular sequence were highly methylated when located within MOSAT repeats. However, such sequences within the genomic context outside of repeats were fully unmethylated. So we conclude that our MCH methylation that we observed by whole genome bisulfite sequencing is not due to some conversion bias, but that it is bona fide DNA methylation signal. Next, what we wanted to look at is where these sequences are located in the genome. And here you can see the breakdown of what we saw. So most of them are found in either intergenic regions or in introns. And you can see two examples, one for each. So this would be the MOSAT sites in the intron and the MOSAT sites in intergenic region between these two genes. And the blue signal is MCH, and this grayish signal is MCG. One other important thing to mention is that these MOSAT repeats are depleted of CpG dinucleotides, so it's likely that MCH within these sites is not just a byproduct of DNMT activity or methyltransferase activity that's targeting CpG dinucleotides. So we can detect MOSAT by both whole genome bisulfite and EM-seq. We find these regions in intergenic context as well as in introns, and we see that they are depleted of CpG dinucleotides. And another very interesting observation what you can see here is that MOSAT MCH appears to be increasing throughout development, unlike CG at these regions. And that's something that we wanted to study in more detail. So to do that, we decided to expand our range of stages, and we reanalyzed a number of publicly available data sets to span the whole embryonic development of zebrafish, including gametes and also other adult tissues. And what Sam saw amazingly was that MCH, that MOSAT MCH, is reprogrammed or remodeled during embryogenesis. And you can see clearly here that it's present in sperm, it's also present in egg and in the zygote. It gets diluted during embryogenesis, during these cleavage stages. It reaches its lowest levels at zygotic genome activation, and then coinciding with gastrulation, it increases, and it is present in brain, liver, and we also have some data showing that it's present in other tissues, so likely all tissues that originate from all embryonic layers. So next we wanted to look at histone modifications and other epigenetic marks that might associate with this motif. And we again utilized published data sets. And what we saw was that the only mark that was enriched on MOSAT MCH sites was K9 trimethylation. And none of the other histone modifications, the active ones or K27 tri, were enriched at MOSAT site. So to explore this in more detail, we remapped these H3K9 trimethylation data that were generated by Mary Gold's lab last year, published in a beautiful study that explored the dynamics of K9 tri. And essentially what we see is, as described in the paper, that this trimethylation signal increases during embryogenesis. However, on a small number of MOSAT sites, it appears to be stable. And you can see here clearly that MOSAT is being reprogrammed. Nevertheless, H3K9 tri remains stable at these regions. And this is a very beautiful example. So you can see very nice correlation in one of these intergenic MOSAT sites. You can see MOSAT slightly increasing, and you can see the overlap is kind of almost perfect with H3K9 tri. And the correlation between MOSAT MCH, at least in stages where both marks are present, so here and here, so in 1.75 hours and 8 hours, so before and after the ZGA, the correlation, the positive correlation between these two marks is pretty strong. So we conclude that MOSAT coincides with K9 tri enrichment. Obviously, K9 tri persists in the absence of MOSAT MCH, so it's unlikely that MOSAT MCH would play a role in the recruitment of heterochromatin. However, we cannot exclude the possibility that H3K9 try helps in recruiting MOSAT back after the ZGA. So that is something that we are currently looking into as well. So finally, we wanted to explore a bit further which DNA methyltransferase could be responsible for this MOSAT MCH signal. And as you can see in Zebrafish, the situation is pretty complicated when it comes to DNMTs, and in particular de novo DNMTs. So apart from the maintenance methyltransferase DNMT1 and the tRNA methyltransferase DNMT2, Zebrafish display 6 de novo methyltransferases. So these were the old names, and more recently these even more confusing names have been adopted. But what's really important here, what I'd like to highlight, is that DNMT3BB2 as well as DNMT3BA have this weird CH, so calpanin homology domain, that is usually associated with actin binding that we have no idea what it does. And it's only specific to teleos DNMTs. So to get a better idea of what to knock out, we first explored the developmental expression profiles of different DNMTs and came up with a couple of good candidates. So as you can see here on the left-hand panel, DNMT3BB2 coincides with its peaks at the ZGA, and DNMT3BA basically tracks perfectly how MCH is coming back after the ZGA. But nevertheless, the first thing that we wanted to do was to exclude the possibility of DNMT1 or maintenance methyltransferase activity playing the role in MCH, as we knew that in mammals it's mostly guided by, it's exclusively guided by DNMT3s. So we designed CRISPR knockouts. So these would be transient CRISPR knockouts or CRISPRs, and we measured DNA methylation by a modified reduced representation bisulfite sequencing approach that allows us to enrich for MOSAT sites. And essentially what we saw, this would be the wild type, and this is CpG, and this is MOSAT, and that's CH methylation. That DNMT1 reduces the CG levels a bit. However, we knew that DNMT1 has this massive maternal contribution, as you can see on this slide. So we also knocked out ERF1, which is the cofactor that actually targets DNMT1, and here we achieved a much better knockout. But as you can see, this is exclusive to MCG methylation. Nevertheless, for both DNMT1, ERF, or DNMT2, the levels are pretty much stable on MOSAT sites. So next we knocked out the de novo DNMTs, and what you can see here is that in the knockout where we combined guides targeting 4 DNMT3Bs as well as the DNMT3BA, here you can see a very significant reduction in MOSAT MCH levels. And all of this has been done a number of times in biological replicates. So we are pretty confident at the moment that DNMT3BA, so one of these weird methyltransferases with a CH domain, would be the main culprit for MOSAT MCH methylation. Even though we're not completely excluding the possibility that the other ones might somewhat participate in it. So we conclude that DNMT3 is the primary MOSAT MCH methylase. We are currently exploring whether this weird CH domain would have a possible function in it. And of course, there will be a lot of redundancy with such a number of de novo DNMTs. So we are currently very carefully designing these experiments. So since this is a reprogramming webinar, I would like to end with a reprogramming figure. And what you can see here is during zebrafish embryogenesis, MCG levels are pretty much stable, somewhere around 24 hours when a number, actually thousands of enhancers are being demethylated, actively demethylated. You can see a slight dip in methylation, and that increases as the development proceeds. But with most at MCAG, you can see that it's reprogrammed very similarly as mammalian MCG and MCH are reprogrammed. So you have them in the gametes, it's practically erased, and then it comes back again in a developmental fashion. So in sum, we uncovered a novel MCH signature in this very peculiar motif, which hasn't been described before. Unlike MCAC and MCAG described in mammals, we find that mossad MCH is found throughout embryogenesis, also in a number of adult tissues, so it's not restricted to pluripotency or the nervous system. It's reprogrammed during embryogenesis, coinciding with zygotic genome activation, and it's associated with heterochromatin. And of course, the million-dollar question is, how does mossad MCH contribute to embryogenesis? How does it contribute to ZGA? What does it actually do? And that's something that we are currently actively investigating. And yeah, of course, it is deposited by DNMT3-BA, the peculiar DNA methyltransferase. So to conclude, I'd like to thank my lab, and in particular, Sam, who did a fantastic job driving this project forward, and Allegra and Michael, who also participated in this story. Alex de Mendoza from Queen Mary University, he helped a lot with genotype correction and motif calling and some DNMT evolutionary comparisons. And I'd like to thank our collaborators from the Radboud University, Michiel Vermeulen and Velin Sequeira, and More from the Andalusian Center of Developmental Biology. They are currently participating in ongoing work, and Ryan Lister for always being able to discuss anything related to DNA methylation and non-canonical DNA methylation. And finally, our funders, and thank you all very much for listening. And with that, I'll leave you to Melanie and her exciting research.
Thank you, Oz, for a wonderful talk. I will just start my presentation. So hello everyone, I'm Melanie, and as I'd first like to start by thanking the Biochemical Society as well as Marnie for the invitation to share my work. So I'm going to switch gears and talk about the mammalian system, in particular mouse early development, and a concept that we're calling epigenetic priming and how this might drive early cell fate decisions that occur in the embryo. So just a quick overview of the key steps that take place in mammalian development. In the mouse, following fertilization of the sperm by the oocyte by the egg, I can't get my mouse to show up, I'm sorry. Okay, you undergo a series of cleavage divisions, which results in a blastocyst structure at E3.5, which is the structure that implants into the uterine wall. So by this stage, you've had zygotic genome activation that's taken place, and you're getting the first cell fate decision has taken place, such that it is just the cells of the inner cell mass, the ICM, which will give rise to the embryo itself. So after this blastocyst structure implants, you undergo gastrulation. So this is when the 3 germ layers really take form, and this is when the cell fates really are starting to be established. So during this time window, you're getting a decrease in developmental potency, but also a lot of transcriptional and epigenetic changes. So at the transcriptional level, the oocyte comes preloaded with maternal RNA stores, which you can see in the green line. And these are rapidly degraded as the zygotic genome is awakened during zygotic genome activation. Coinciding with this, there's changes in the DNA methylation landscape. So in mammalians, DNA methylation occurs mostly at the CG context, and most cells are about 80% of their CGs are methylated. However, in the pre-implantation embryo, you get this massive wave of global loss in DNA methylation. And DNA methylation really only comes back up again during gastrulation as sulfates are being set up. At the same time, another epigenetic structure that I'll talk to and introduce in a few slides, bivalency, this is also lost, is absent in the pre-implantation embryo and really becomes established at the early stages of development. And so the question we have is how the epigenome really facilitates these early embryonic sulfate transitions, and can it in a way preempt these sulfate decisions and set the cell up for enabling it to be able to make these transitions. So this is a concept that we're calling epigenetic priming, which I'll just illustrate here with this cartoon. So the idea is that a priming factor showed by the blue balls may be sitting at pluripotent genes, at lineage-specific genes in pluripotent cells, shown in the upper left quadrant. And so by sitting at the promoters of these genes, it can be preventing repressive DNA methylation from coming in, keeping the chromatin structure open by repelling repressive epigenetic modifiers, shown in red, or by recruiting active epigenetic modifiers, shown in green. And in this way, it's keeping the chromatin state, epigenetic state of these genes primed and malleable so that upon differentiation, they can then be activated, even in the absence of the original epigenetic priming factor. If you lack the priming factor, which you can see in the bottom panels, then you can have repressive DNA methylation coming in. The repressing epigenetic modifiers may be able to be recruited to these genes and lock the structure such that upon differentiation, these genes are now no longer able to be effectively activated. So there is a good example that we've known about for several years now of epigenetic priming, and this is the context of bivalent chromatin. So this is the co-occurrence of both active and inactive histone modifications on the same nucleosomes. It occurs primarily in undifferentiated cells at key developmental gene promoters. So you have co-occurrence of the active H3K4 trimethylation mark, which is deposited by the compass complex, and the inactive H3K27 trimethylation mark, which is deposited by polycomb. And the idea is that upon differentiation, these poised structures are resolved into either a repressed H3K27 containing or an active H3K4 trimethylation containing structure as sulfate decisions are made. However, we know little about how the bivalent chromatin is actually specifically targeted to these key developmental gene promoters. So the proteins I want to talk to you today about that we have discovered as key epigenetic priming factors are called developmental pluripotency associated 2 and 4, or DPPA2 and 4. These are quite small heterodimerizing proteins. They're about 300 amino acids in length, and they contain both a Sap domain that binds DNA as well as a C-terminal domain that binds histone H3. They have no known enzymatic activity that we have found. And what's been quite perplexing is that the knockout mice for these proteins survive embryogenesis but die shortly after birth. And this has been quite a conundrum in the field because these proteins are exclusively expressed in early embryos. So if you can see in the bottom right graph, their expression is really limited to pre-implantation development. And by E6.5, when gastrulation starts, their expression is absent. And they're no longer expressed throughout the whole of rest of embryogenesis into adulthood, except for in the germline. So this actually makes them ideal priming factors in that they are expressed at a time, but their phenotype is seen at a later developmental time point. We came across these proteins in a screen we did looking for regulators of zygotic genome activation. And in this paper that I don't have time to share today, was published last year, we found that DPPA2 and 4 act together by binding the promoter of a transcription factor called DUX, promoting its expression. And DUX is a key transcription factor for zygotic genome activation genes. And during this study, we also noticed that DPPA2 and 4 also bind non-ZGA genes. And it's this that we decided to follow up on further. So this is them binding non-ZGA genes. In particular, because when we made double knockout embryonic stem cells for DPPA2 and 4 and subjected them to pluripotent to differentiation assays, we saw quite a striking phenotype. So if you differentiate wild-type embryonic stem cells over 9 days of embryo body culture, usually we see downregulation of pluripotency genes by about day 4 of differentiation. And this coincides with the upregulation of mesoderm, endoderm, and ectoderm genes at about day 4, as you can see in the left panels of the heat map. However, if you take the double knockout embryonic stem cells and put them through the same differentiation assay, you can see that the pluripotency genes take until day 7 to be downregulated, whereas the mesoderm, endoderm, and ectoderm genes are now also being upregulated at a later time at day 7. You can also see this in a principal component analysis where we take the whole transcriptome and cluster the cells based on how similar they are in the transcription. And when you do this, you see that the double knockout cells at day 7 and day 9, which is shown by the pink and purple diamonds, more closely resemble the wild-type cells at day 4, which are shown by the red circles, than the wild-type cells at day 7 and day 9, which are at the end of the arrow. And so this really points that these cells, although they have no problems in their pluripotent state, they're having problems differentiating. So when we looked at where these cells are binding throughout the genome, we noticed that they are highly enriched at the promoters of active and bivalent genes. So on the left, I'm showing you an example where you can see some active gene promoters highlighted by the green bar and some bivalent gene promoters highlighted by the yellow bar. And the tracks, you can see the DPPA2 and DPPA4 binding in the blue and the purple, and the K3 and the K27 trimethylation in the green and the red. And you can see nice enrichment at these promoters. And when you actually do genome-wide analysis and look for the enrichment, you can see that these genes are really enriched at active and bivalent promoters. So this prompted us to look at the epigenetic state of the embryonic stem cells when you have removed DPPA2 and 4. So on the top, I'm showing H3K4 trimethylation, and the bottom H3K27 trimethylation. This is whole genome by ChIP-seq. And I'm showing here the peaks, and you can see that in the H3K4 trimethylation in the knockout cells, you're losing a large number of peaks which are overlapping with DPPA4 peaks in purple, as well as promoters shown in orange. You're also losing H3K27 peaks, although to a lesser extent. And indeed, these peaks are overlapping each other and actually are co-occurring at a lot of bivalent genes. So if we zoom in particularly at the bivalent gene promoters, we can categorize them into 4 different categories, or 3 different categories, sorry. We have those in orange, shown in the top row, which have lost both H3K4 trimethylation, H3K27 trimethylation, as well as the epigenetic machinery responsible for depositing these modifications. And we're terming these bivalent genes DPPA-dependent bivalent gene promoters. We also have a second category of promoters which are sensitive to loss of DPPA2 and 4. So these are losing H3K4 trimethylation, but the H3K27 trimethylation remains unchanged. And lastly, we have a group of promoters which are independent of DPPA2 and 4. So these are remaining largely unchanged. And what you might appreciate from this graph, and we've done some machine learning analysis as well to support this, is that the main feature that distinguishes these 3 categories of bivalent genes is the original amount of H3K4 trimethylation at the genes. So DPPA2 dependent bivalent genes have fairly low levels of H3K4 to start with, whereas the sensitive and the independent ones have higher levels of H3K4 trimethylation. And when we look specifically at these different bivalent gene categories, of their behavior during differentiation, you can see that all three categories in wild-type cells usually become upregulated upon differentiation. However, the DPPA2 dependent gene promoters shown in the orange, in the knockout cells, they are now no longer able to be activated upon differentiation. So when we next ask, well, what is keeping them repressed? We turn to DNA methylation, as this is a well-known mechanism for repressing gene expression in mammalian cells. We did a whole genome bisulfite sequencing, and you can see in the left plot that most of the genome remains largely unaltered. However, we have several hundred regions of the genome which are gaining DNA methylation, and these are overlapping gene bodies shown in pink and promoters shown in orange. And you can see the quantification of these in the pie chart. And indeed, when we look specifically at the dependent gene promoters shown in orange in the right plot, you can see that these are the regions that are gaining DNA methylation in the knockout cells. And we think that this is a subsequent event to the loss of bivalency and is what is keeping them silent during differentiation. So lastly, we wanted to develop a system to look into the dynamics and the recovery, whether or not we can rescue this phenotype. So to do this, we turn to an inducible shRNA system whereby addition of DOX enables the expression of shRNAs targeting DPPA2 or DPPA4. And you can see in the Western blot that by 7 days of treating with DOX, you have lost DPPA2 and DPPA4. What's quite nice about this system is you can then wash DOX out and allow the cells to recover. DPPA2 and 4 levels return, and you can ask, well, what is the epigenetic state in the recovery cells 2 weeks after depletion? So when we do this, you can see in the left-hand panels are PCAs. So we start with the untreated cells are shown in the gray circles and squares. Addition of DOX, which leads to the downregulation of DPPA2 and 4. In the pink cells, you can see these are very different in their epigenetic profiles for both K4 and K27. However, now when we allow DPPA2 and 4 to return to the cells, in the recovery cells in blue, you can see that they now closely resemble the original untreated cells. So we are restoring this altered epigenetic state. We look at the regions of the genomes that are changing in H3K4 in the top or H3K27 in the bottom. You can see that between untreated and DOX-treated or DPPA depleted cells, you can see that we're losing a large number of H3K4 trimethylation peaks. H3K27 doesn't change as much. And indeed, if you look specifically at the DPPA-dependent genes in the right panel, highlighted by orange, you can see that these are the genes promoters which are losing H3K4 trimethylation in the DOX-treated or DPPA-depleted cells, and they are now being restored in the recovery cells. So this suggests that DPPAs are sufficient for actually targeting this bivalent structure to these genes. And lastly, we also look to see, well, what's happening at the DNA methylation at these sites in this inducible recovery system. And we did this using amplicon bisulfite sequencing. And if you look here, this is a list of DPPA-dependent genes. The top 2 rows are control regions. And you can see in the untreated DOX cells, they have relatively low levels of DNA methylation. This increases in the cells treated with DOX, which have depleted DPPA2 and 4, and is now lost again in the recovery cells. This again points at DPPA2 and 4 targeting the bivalent structure, and really the loss of them is the DNA methylation coming in subsequently. So just to summarize this study with an example gene. This is a DPPA-dependent gene. You can see I've highlighted the promoter region with this pale orange box. You can see the top in the top tracks, you see binding of DPPA2 and 4 at the promoter of this gene. In wild-type cells, there is a nice peak of H3K4 and H3K27 trimethylation, and this is lost in the knockout cells, completely absent. Coincidentally, we also have a loss in accessibility, and we have a gain in DNA methylation shown by the long lines from wild-type cells shown in gray to knockout cells shown in green. And subsequently, upon differentiation, you can see these are transcription tracks from days 4 of differentiation in wild-type cells on the top row. You can see that there is transcription. This is lost in the knockout cells on the bottom row. So this led us to our model, whereby there are several classes of bivalent genes, the DPPA-independent genes and DPPA-dependent genes. And the DPPA-dependent genes shown in the bottom row, DPPA2 and 4 are present and really required to recruit polycomb and trithorax and maintain this active and inactive poised bivalent structure. In the absence of DPPA2 and 4, you fail to recruit polycomb and trithorax, you lose this bivalent structure, and subsequently DNA methylation comes in, represses the genes, such that upon differentiation, these genes are now no longer able to be expressed. And so we think that DPPA2 and 4 act in this way as epigenetic priming factors and really important for multilineage commitment. And they do this by targeting bivalency to a subset of bivalent promoters, preventing repressive DNA methylation so that these genes are activated efficiently during differentiation. And we think that this temporal uncoupling of when the proteins are expressed and the phenotype of the genes that they regulate may explain the pre-weaning lethality of the knockout mice. And we think that there may be actually a more general role for DPPA2 and 4 in promoting epigenetic plasticity in general and facilitating cell fate transitions. So with that, I'd like to thank the people in the work in the lab who have helped with this project, in particular Alid, Crystal, Elisa, and Malush, as well as the collaborators Clive Dos Santos and Masashi Narita at COUK. And just to let you all know that I'll soon be starting my lab at Peter Mac in Australia, so looking at epigenetic priming and development and cancer. So if you're interested, there'll be future opportunities here. And thank you for your attention, and I guess we'll pass back to Mani for the question and answer session.
Thank you so much, Melanie, and also thank you, Oz, for your fantastic talks. So everybody, please remember to send in your questions. I'll start asking the ones that I can see already. So the first one for Oz says, does the ablation of DNMT3BA and thus MCH result in an increase in MCG at the same regions? And this comes from Aitan Zlotorinsky.
Yeah, we haven't seen that. So it appears to be specific to MCH, not to MCG.
Excellent. Okay, so for Melanie, Frank Schubert says very clear data. Does DPPA2 and 4 dependence identify genes that are critical for cell fate decisions, or rather genes involved in subsequent differentiation processes?
Yeah, it's a great question. So they tend to be genes that are involved in actually the cell fate decisions themselves, along with a lot of bivalent genes. When we do gene ontology, we don't really pick much up. Although if we look at the phenotype of the knockout mice that have been generated for these dependent genes, a lot of them have very clear developmental phenotypes, but at later stages, so later stages of embryonic development or pre-weaning just after birth.
There's another question for Melanie from Samuel Ross. Just a clarification, I think, are DPPA2 and 4, are they similar to Stella, or is it just a generic name?
Yeah, it's a good question, actually. So there are 5 DPPAs, and they were named in a screen looking for proteins which have similar expression pattern to OCT4. So they were based on their expression patterns, not necessarily their structure. Coincidentally, DPPA2 and 4 are very similar. DPPA3, which is also known as Stella, is fairly similar, but as far as we know, doesn't act in this sort of manner.
Okay, I'm just trying to work out who this one's for. It just says, many thanks for these great talks. It's from Susan Dietrich. How come if differentiation is globally inhibited, the phenotypes appear so late? So cardiomyocytes you need very early, for example. So this is obviously Melanie. Sorry, I worked it out after reading.
Yeah, and that's something that we're still trying to understand, actually. So one idea is that you can sort of think about cell fate transitions as activation barriers. So if you you might still be able to get through eventually. So if you saw in the heat maps, eventually the genes get activated upon differentiation, but they're just delayed, so it's just not as efficient at doing it. So you might be able to sort of start making the tissues, just not as efficiently, and they're not working as well. And so that's why you might be able to get through all of development to birth. So lay out the rough body plans, but not perfect body plans. And it's really at birth when the mice need to start breathing and eating and so on, that's when you get the lethality. So actually they die just after birth from lung defects, which obviously makes sense because that's the first time they need to use their lungs. So we're following this up at the moment. We've made knockout mouse models, and we're going in and profiling them at earlier stages to see how their epigenetic state looks, and sort of later stages to see how they look phenotypically. But that's the idea that we have at the moment.
Maybe a related question to that from me. So there are many different epigenetic regulators. When you knock them out or mutate them, you get quite a wide variance. So maybe you haven't changed overall the average, but you have this huge variance, and it sort of looks like, well, something goes wrong, and it just depends on what goes wrong first. Do you see that in these mice, or is it they're all dying of a lung defect, or do some die in late gestation and they're kind of dropping off
we don't really see much of a dropout earlier. We haven't got enough numbers now to really have a proper statistics, but most of them seem to be making it through. The litter sizes seem fairly similar. Occasionally you get some mice that survive, and while they're lighter, they're smaller than their litter mates, the wild type litter mates, they catch up eventually. So this does, there is a heterogeneity in the litters themselves and amongst the different litters. And so it's not an all or nothing, black and white sort of thing. So it does point as, an efficiency, or these proteins are really, improving the, improving the efficiency of making these decisions rather than being the key, the gate holder for it. If that makes sense, yeah.
I'm going to ask one to Oz now, if that's okay. So I was wondering, Oz, do you know, or do you postulate about any of the particular binding factors for the MCH methylation in MOSAT? So I guess in mammals, MECP2, which classically I have thought of as a CG binding protein, has now been shown to bind CA. So do you think there could be factors like that we might have previously thought had maybe a more important role at another site that might be binding these?
Yeah, so we are currently looking into that. But it appears, and that's also based on the motif, some people probably picked it up, it very strongly resembles the OCT4 motif. And actually it appears also to coincide with sites of OCT4 binding during embryogenesis. And we have some preliminary data that MOSAT and MCH, when methylated, has high affinity for OCT4. So how it all plays into the context of embryonic development is something that we're currently looking into, but we think that what might be happening is that some of these sites might actually serve as sinks or reservoirs for transcription factors, which then get redistributed upon the ZGA. And recently, Julia Horsfield showed something similar with cohesin as well. Yeah. So it is possible it participates in a similar mechanism.
Yeah, and it's a more efficient way of doing things early in development. Okay, there are a few more questions. So the next one is from Grant Mitchell. It says, 2 nice talks. To translate to humans, is the DPPA phenotype detectable in fetuses, and would it appear in humans by prenatal ultrasound, so intrauterine growth restriction, for example, or methylation?
Yes, that's a great question. I have looked into it, and there's actually no cases of humans that have mutations in DPPA2 and 4. Like, we don't know what the mutations, but loss of the DPPA2 and 4 locus. So whether that's just because they don't have a phenotype, or whether that's because these humans don't exist, of course we don't know. We do have some ongoing work in the lab looking in human embryonic stem cells to get at some of these types of questions, because it is quite interesting whether or not the same thing is also happening in the human system. That's a good question, yeah.
What about, so a separate way to think about relating it to humans would be to say loss of bivalency and a gain in DNA methylation sounds distinctly like what happens in tumours. CG islands anyway. So do you see a change in them in the context of cancer? So they're not normally expressed then. Do you see them being aberrantly expressed or something weird going on?
Yeah, no, exactly. And that's something that I'm planning to follow up on in my new lab, is that there's quite a lot of cancers that upregulate DPPA2 and 4. It's thought, so these genes are regulated themselves by DNA methylation. And so we think their upregulation, it's not through copy number amplifications, but it's actually through changes in the methylation at the promoters that lead to the upregulation of these genes in a lot of different cancers. We don't know the significance of this yet, but my hypothesis is that their presence and upregulation promotes this epigenetic plasticity in the cancer cells, which you can see in the altered epigenetic profile of the cancer cells. And this might actually be contributing to tumorigenesis or metastasis when the cells actually have to change what they're doing and, you know, become more malignant, for example. But yeah, it's something that, stay tuned. Hopefully we'll have an answer sometime soon.
Yeah. There are two questions from Angel Yang, so they kind of follow on. Like I'll ask them both at the same time. The first says, how do DPPA2 and 4 know where to be recruited to? And do you think that they could be used to enhance pluripotency in some way artificially?
Yeah, so they bind CG-rich regions of the genome. So they like promoters, CG-rich promoters. They're actually found at 2/3 of promoters. So we don't know exactly why only some of the bivalent promoters are affected, whereas other bivalent promoters that have DPPA2 and 4 binding are unaltered. I think it's to do with levels of K4 and transcription of these genes, which are able to maintain them in a state that you lose the initiator and they're self-sustaining. But we still need more data on that. So
the acetylation, the amount of acetylation they have there rather than K4 try or K27 try?
Nothing that's really stood out. I don't think we've gone through comprehensively. We've done motif analysis, but it just get GC-rich sequences because they like GC-rich regions. And for enhancing pluripotency, there's a really nice study that was done out of the US looking at DPPA2 and 4 in iPS cell reprogramming. And actually there, by overexpressing DPPA2 and 4, you really greatly enhance iPS reprogramming. Up to 80% of cells can make it. So there is certainly something about cell fate transitions in general, not just, you know, the ones at bivalent genes and gastrulation, but also ZGA reprogramming and so on. So yeah.
Cool. I have one more for Oz. Can you in your expression analysis, can you do expression analysis at the same time as your CH methylation analysis? Perhaps not during the early stage where K9 methylation seems to take over or be retained while the CH is being reprogrammed, but later can you correlate the amount of CH methylation and the context, so where they are in the gene and whether that gene is being expressed or whether they're in the surrounding genes in an intergenic region? Can you kind of
so we looked into that, and yeah, and unfortunately because of the restrictions on the time I didn't put it in, but we don't think that it correlates at all. Actually, interestingly and similar to mammals, most of these most at MCH sites, they're found in neuronal genes. So even though the developmental increase has nothing to do with these genes because they get switched later on, there must be something that attracts most at MCH and the whole neural story. I don't know if it is because neural genes are longer, so they have a higher probability of having you know, such a massive repeat cluster, or if there's something else, or if there's something related to the 3D structure, but there's definitely no correlation in terms of most at MCH and gene expression, at least not of that very gene where it sits in.
The other thing that I thought was interesting was that you had the CH. The CH is where you are CG poor, and it's CT. So that you know, is there any? Can you go back in evolutionary time and ask when that repeat landed in those places and what it was like beforehand to try and see if there's any relationship?
Yes, so we looked a bit into it. It appears that it's specific to zebrafish and carps, and there's some data, published data, that found similar repeats in the Asian sea bass. But yeah, that's something that we would like to look into. But it is it has to be done in a comprehensive manner, and it's not so trivial actually.
No, exactly. I'm sure it's very hard. Yeah, it does sound.
But yeah, it would be interesting, definitely. Yeah.
So there's one final question, and this is for Oz, and then we'll wrap it up. So Susan Dietrich says, is this MOSA enrichment because of primary neurogenesis in amania and mania? You might understand that better than I do.
Yeah, not sure really, but it's, yeah, I don't think that it correlates really with neurogenesis, because as I said, this enrichment that we're seeing starts happening even before that.
Yep.
So we think it has something to do with general regulation at the ZGA, and the fact that these sites reside in neuronal genes. It's likely because of their length, or because of some other, I guess, structural concept that they would fit nicely in that environment.
Okay, so it just remains to thank both of our speakers. I'm so very grateful that you were able to join us tonight, and everybody who came along and asked questions and listened. So we're going to be running an embryogenesis collection in the Biochemical Society transactions, and that can be visited at the journal website for more information. If any of you that's watching out there would like to suggest key topics that we should commission around, so commissioning short reviews, or if you'd like to submit a mini review to the collection for consideration, please contact editorial@portlandpress.com. And we're going to also welcome suggestions for future topics and speakers to feature in this Biochemistry Focus webinar series. So if you've got a good idea for a webinar, then please do submit a proposal for an upcoming webinar. The next one is going to be held next Friday, the 11th of September at 3 p.m. UK time, I think, or 1500. And it's on how to deliver biomolecular science honours research projects with limited lab bench time, which is relevant to many of us that are supervisors. And this is part 2 in that webinar series, where Dr. Aisha Divan, an associate professor in the School of Molecular and Cellular Biology, and Dr. Vas Panambilan, a reader in the Human Disease Biology at University of Leeds, will talk about their experiences of running non-traditional research projects under the constraints of limited access to the lab. So you can register for that one and find out more information about the upcoming webinars, and of course propose your own webinar at www.biochemistry.org.webinars. Thank you very much.